In the effects of growth inhibitors, we have studied that such growth inhibiting agent induces a condition of dormancy. In this section, we will understand that what is seed dormancy. Seed dormancy is considered as a condition in which the seed growth is arrested even under the favorable condition. When such seed growth gets arrested, the degradation of the plant occurs, resulting in the decreasing growth. For this, various factors are responsible and various types of seed dormancy occur. In this, some seeds can remain dormant for months, for a few months, or even years. Thus, it is not definite or constant time period of dormancy for any plant. Since the time of seed dormancy can vary in different plant species in a different manner. Now, there are basically four types of seed dormancy. First, exogenous dormancy. Second comes endogenous dormancy. Third, combinational dormancy. And fourth, secondary dormancy. So now let us start with our first type, that is exogenous dormancy. Exogenous dormancy is a condition of dormancy which arises due to the conditions that are present outside the embryo. Such a type of dormancy generally arises because of mainly three parameters, or they can be of three types. First, physical endogenous dormancy, in which the condition arises due to the impermeability of water or gases to the cells outside the embryo. Second condition is known as mechanical endogenous. In mechanical exogenous dormancy, the seed coat develops hard covering around the seed, due to which a dormancy state arises. The third type is known as chemical, exogenous dormancy. In this type, induction of various growth promoters or inhibitors, that is growth regulators, occurs outside the embryo. Then comes the second type of dormancy, endogenous dormancy. This type of dormancy is entirely opposite to that of the previous one, because in this, conditions arising within the embryo causes dormancy of this type. They are also of basically three types. First, physiological. In this type of endogenous dormancy, inhibition of seed germination and seed growth occurs. The second type of endogenous dormancy is morphological type. This type of dormancy occurs when the embryo differentiation does not occur, at the time of fruit ripening, which causes undifferentiated or underdeveloped embryo. The third type is a combination of the above two, that is combined dormancy, which is also known as morphophysiological dormancy. Because it is formed by the combination of physiological and morphological dormancy. This was all about the endogenous dormancy. Then comes the third type of dormancy, that is combinational dormancy. Definitely the name suggests that it is formed by the combination of two types. That is, the condition arised by the combination of exogenous and endogenous the physical type of dormancy within exogenous and physiological type of dormancy in endogenous. Together combines to form combinational dormancy. The fourth and last type of dormancy is secondary dormancy. This type of dormancy occurs when the condition unfavorable for the seed germination like decreased temperature, that affects the secondary growth, leads to the condition of secondary dormancy. This we have all studied about the types of dormancy. Now let us study about the causes and its removal. Now after studying about the types of seed dormancy, let us understand that what are the various causes that leads to seed dormancy. The first cause is underdeveloped or physiologically immature embryo leads to such a condition. Second, mechanically hard strong and rigid seed coat development develops water impermeability as a result of which it inhibits the seed germination. The third and the most important cause of seed dormancy is induction of growth inhibitors like ABA, that is abscisic acid, which inhibits seed germination. Because production of abscisic acid leads to the inhibition of transcription of genes as a result. The proper enzymes that are needed for seed germination are not produced. Due to which the seed germination gets inhibited, and as a result the seed dormancy condition arises. After studying the causes, let us understand that how can seed dormancy be removed. As I studied earlier, the condition of seed dormancy arises only when the seed contains a large amount of abscisic acid, and therefore it is necessary to decrease the level of abscisic acid. But how this can be done? Growth regulator gibberellin induces seed germination, which has effect exactly opposite to that of abscisic acid, and therefore increasing the concentration of gibberellin than the abscisic acid or ABA within the seed makes it active by removing the dormancy. Such an increased concentration of gibberellin and decreased concentration of ABA can be brought in various manners, which are artificially induced. Let us study some of these methods. The first method is that seeds that are dormant can be scrapped lightly with the sandpaper or with other chemicals which makes the seed permeable to water and thereby help in the germination process. Other method is the soaked seeds should be placed in an oxygen containing environment for a definite period with a specific temperature that can be either higher or lower depending on the condition, removes the dormancy and thereby provides the path for the successive seed germination. In this way, the removal of seed dormancy can be done, which thereby regulates and induces the seed germination process further.